Okay, next up, from Guadalajara, Mexico, we have Narval. Presenting for Narval is Dr. Jose Luis Nuno and Alejandra Nuno. Give them a round of applause and bring them on out.
Hi, I'm Jose Luis Nuno, CEO and founder of Narval. I am not a morning person, but after my first coffee, I am ready to solve diseases and change the world. $600 million live with diabetes. 37% will develop diabetic retinopathy, an eye condition that causes vision loss and blindness. The available treatment is a $2,000 monthly injection of an antibody drug in the eye. This is complex, risky for the patient, and expensive, but cannot be done otherwise with the therapeutic alternatives on the market. No matter if you suffer from a respiratory disease, cancer, or a digestive disorder, chances are that you are going to be treated with an antibody drug that costs thousands per dose and can only be delivered by injection. Antibody drugs are a $200 billion market and fundamental to treat diseases, but these are not easy to use or affordable. Antibody drugs are made with monoclonal antibodies, which have significant challenges like their incapability to cross through most tissue barriers on the body, lack of stability out of cold chain, and high manufacturing costs. This limits their use into a very narrow set of applications, making impossible the development of new antibody drugs for hundreds of diseases that affect most of the global population. At Narval, we are solving this problem, designing synthetic antibody mimetic proteins to replace monoclonal antibodies and novel first-in-class therapeutics. Our goal is to create antibody drugs that, until now, have been impossible to develop. Our synthetic antibody mimetic protein platform, or AMP for short, uses CDR3 regions from single-domain shark antibodies fused to a synthetic conoprotein scaffold from a sea snail. Our AMP platform uses molecular and artificial intelligence processes, eliminating the use of in vivo, transgenic, and cellular models. Our AMPs are 40 times smaller than a monoclonal antibody, but with the same biological functions. Our AMPs can cross through tissue barriers, generate limited immune reactions even with no humanization, can target very small and hard-to-reach antigens, and are heat-resistant. These advantages gives our AMP platform the ability to develop antibody drugs that have been impossible to develop, like antibody drugs in aerosol, in eye drops, viral delivery, or transdermal release. Our mission is to make antibody drugs as affordable, accessible, and easy to use as common pharmaceutical drugs. Now, let's move to the overhead camera, please. We are going to show you what our AMPs are capable of. Alejandro will show the effect of an AMP over a respiratory virus on a long cell culture. This experiment takes several hours, so today we are going to see only the outcome. Now, move to the demo, please. The microscope. Thank you. Here we can see healthy lung cells, which are representative of someone with no respiratory infection. To these healthy lung cells, we added a sample of the respiratory virus. The virus infected the cells for replication, destroying the cell structure on their way out after a few hours of incubation. On a second sample, we added the virus and our AMP. The antiviral AMP neutralized the virus, blocking its ability to infect the cells and to replicate, protecting the cells and therefore the patient lungs from the viral attack. Now, back to the presentation, please. We designed our AMPs from validated targets, focusing our core activities on development, validation, and preclinical testing of our AMPs. Then we out-license our AMPs to pharmaceutical companies, generating revenue from upfront payments, clinical phase, and approval milestones, and then royalties over sales of the marketed products. Our goal, our vision, is to create the largest portfolio of antibody drugs for unserved therapeutic needs. Our first AMP candidate, AMP1 for diabetic retinopathy, is going into clinical phase next year, and we plan to license it on early 25. Our AMP1 on in vivo testing shown proven capabilities for eye tissue penetration and therapeutic activity on the back of the eye, including antivascularization and a decrease of endothelial cell count of the macula. This means that you will be able to take your treatment in simple eye drops at home, no need for an eye injection. Also, we are working on our first leads for respiratory diseases. Our AMP4 shown full neutralization capability of the COVID-19 virus on lung cell culture at nanomolar level. This is similar to FDA-approved COVID-19 antibody drugs on the market. For me and Alejandro, this is our third startup working together for almost 17 years in biotech. First, we launched a pharmaceutical product for animal health in 14 countries in partnership with a global big pharma. Then we launched a new diagnostic technology using shark antibodies, microfluidics, and artificial intelligence. Now at Narval, we are joining with our academic co-founder, Dr. Alexei Lisea, a world-renowned scientist with deep experience on marine protein engineering and also a former founder. Today, we are launching Narval. Come and join us bringing down the limits on healthcare. We are Narval, and we are the future of antibody drugs today.
Wow, give them a round of applause. I think that's the first time we've ever looked at cells on this stage before live, so thank you for that. Okay, who wants to go first? All right, Bo?
I know the anti-VEGF market. I know that the antibodies work. A lot of them are coming off patent, and you all have to inject them. The data on how it passes to the back of the eye, to the retina, it went by pretty fast on the slides, so I want to understand that more. But my key question is, can you tell us about who can manufacture shark antibodies today, and what do you need to do in a system to be able to manufacture your drug? So how do you making your drug, even for the clinical procedures you're going to start next year?
Can you guys take one step closer to me?
Yeah.
Get in the light there. Thank you.
Thank you. So something really important is this is not the full shark antibody. So what we do is we select the shark antibody from a library, and then we just isolate the CDR3 and fuse it to the scaffold. So it's just part of a shark antibody. So these AMPs are completely synthetic. So they are manufactured by synthesis, like a peptide. So we are not using bacterial cells.
You're not, or CHO cells or anything like that? Okay.
Yep.
Yeah. Can you speak a little more to the ability to get to the back of the eye there by just an eyedropper treatment?
You want to do it? No. Okay, so can you repeat the question, please?
Yeah, so the idea is we don't inject to the back of the eye, right? We put it in the eye and it travels to the back of the eye. And then you want to have a therapeutic window once there. Can you talk a little bit more there about what that looks like?
Okay, so what we have shown already on testing on animals, around the third hour after the application of the eye drop with the anti-VEGF AMP, we see already the AMP inside the eye. So we measure that. So for example, 10 milligrams of the solution on the eye, we can measure this up to like 24 hours after the application. So what happens is travel through the tissues, and then it gets to the back of the eye, and then it decreases the vascularization, for example, in this case, on the area, and also the decrease on endothelial cell count. So this helps to maintain, it's doing the same thing that the monoclonal antibody, but the application way is different.
Mark?
Can you talk a little bit about how insurers and carriers will play into your commercialization model going forward?
Oh, sorry, I can't hear.
Sorry, can you speak a little bit to, from a commercialization perspective, how do you need kind of insurers and carriers to play into this going forward versus, how much is out of pocket from the consumer versus covered by insurance, and how do you imagine that mix to kind of grow or stay stagnant going forward?
Okay, in this case, well, our business model is to work with the pharma company. The pharma company is the one doing the commercialization of the molecule. So, but it will work the same way as any other monoclonal. So it's the same process, it's the same commercialization strategy, etc. The thing that we are substituting the use of a monoclonal which is injected with an AMP that goes into eye drops, but the model is the same one.
I think I might have missed it, but can you speak to some of the efficacy data that you have? I'm sure you've done some testing on this as well. If you could share some of that, and then what the response has been from pharmaceutical companies to that data and to your designs, especially on the AI aspect of what you're doing to calculate all of these outcomes.
Okay, so let's start with the clinical data. The most important part of the preclinical testing that we did was on animals. So we already have testing on the migration of the AMP inside the eye and the antivascularization processes and the therapeutic processes. So what happened is we see the AMP 3 hours after application already inside the eye, and we see a decrease of, it was like 40% of the endothelial cell count inside of the eye, like around the third day after application.
40% after the third day
Yeah.
after application.
Yes.
But the action started within 0 to 3 hours, and you saw that immediately.
No, we didn't measure so we didn't measure after the 3 hours that happened. So no, we were measuring like every single day, so every 24 hours. So we don't have data for the first like 3 hours or minutes or so. And in terms of what is the interest from a pharmaceutical company, we have been already meeting with pharmaceutical companies. We're already closing our first contract with a pharmaceutical company for a disease that we cannot disclose right now, but there's already a contracting process.
What is that contract for? Is it for your technology, or is it for your actual output?
No.
Of
what we sell or what we license is the molecule.
Okay.
Already tested, developed, etc. So we transfer the molecule already pre-IND. So ready to go into phase 1, first in humans. Yeah. So we're already closing the first contract, and there's already like other 4 pharmaceutical companies ready to sign.
Sarah, Libby?
I was going to go in a different direction and just ask, you guys have founded something together before, curious kind of lessons learned about yourselves as complementary co-founders, or about go-to-market or whatever, lessons learned from that experience that you're bringing into this venture.
You want to start?
Yeah,
You
well,
have 30 seconds.
okay, very fast. All the adversity that we have found has taught us more than we have learned from school so that's something I have learned.
Something important for us is the importance about the network that you're building. Right now we have a very big network in the pharma industry, et cetera, because it's already your sort of startup. So we found that's very important. The team that you're building with you,
Yeah.
and the relationship that we have already with pharma companies and research centers, et cetera.
Thank you very much. Give them a round of applause.